Okay, welcome back Science 10s to our next lesson, which is 3.7, and we're going to be talking about transport across membranes.
So from what we saw from last lesson was if we have a high concentration of chemicals on the outside compared to a low on the inside, that these are going to naturally flow in by diffusion. Now this could be the same for things exiting the cell. If we have a whole bunch of chemicals inside the cell and not much outside the cell, it will flow out naturally until it becomes even. But this is only going to happen for really small molecules that can pass through that phospholipid bilayer. So something really small like oxygen might be able to pass in like those yellow molecules, and CO2 being able to pass out like those blue molecules. But what about like some other things? Like what about if you had like a really large molecule? How would that get in? Say if you needed it, say it was something that was a nutrient for the cell. And then if it does let in big things, how can it control what enters and what exits? What's the good things to let in? What's the good things to let out? How does it have a control over that whole process? So that's what we're going to be talking about in today's lesson. And off the start, we're still going to be talking about passive transport, and this is going to be a type of diffusion, just as we saw before. But now we're going to add on to it that it's going to be facilitated. And pretty much this just means that it's going to be controlled diffusion. We're going to be able to control when diffusion happens and when it doesn't. So say we had a high concentration on the outside, they're too big to flow through. It wants to flow in, to go from high concentration to low concentration, but we're just going to be able to control and kind of with a gate open up to see when things should flow through and when they shouldn't. So we're going to look at two proteins that do this, and they're called transport proteins. The one on the left here is called a channel protein, and the one on the right is a carrier protein. We'll look at the channel protein first, and it's when some big molecules, like these blue ones, come up to it, and they can't pass through. They can't fit through the phospholipid bilayer. But if this is something that the cell needs to take in, it has the control to be able to open up this channel, make it wider so big molecules can flow through, and then they can just go through by the rate of diffusion, by flowing in like that. That would be something that we want to diffuse inside of the cell. But if that blue one comes over to a carrier protein, it doesn't fit the mold which it shows right here for the carrier protein. It's much more specific on what it lets in. So it needs something to fit that exact type of molecule that it's shaped for. So the blue one wouldn't work, but this purple one would fit. When it goes into that carrier protein, it's going to fit right into there. It's able to close, it fits perfectly, and then opens up the other direction and it passes through. So it's much more specific in what it allows through, but can still handle those bigger molecules that don't just go through the phospholipid bilayer. So those two things important to remember: channel proteins and carrier proteins are two types of transport proteins and how they work. Now that was still passive transport, and next we're going to look at active transport. We can think of active as like an active person or like running or doing exercise, because it requires energy to do that exercise, and it requires energy to do active transport and put things inside the cell that might not naturally flow there. So this is using energy to reverse the direction that things would normally flow. So say that if we had a high concentration of these blue chemicals on the outside and only a couple on the inside, just naturally, say if they're small enough to pass through, they would go in this direction. They would diffuse from high concentration to low concentration. But in active transport, we're going to force it to go the opposite way. We're going to push those from low concentration to high. So they started from low concentration and they went to high concentration. This is the opposite of what it would normally do. So to push it back in the opposite direction requires energy, and that's active transport, because it requires energy to push them or pump them back to the other side. This could happen through a channel protein as well. So we have a whole bunch of big green molecules over there, only two through here. Again, it would want to flow naturally this way, but we're going to pump it in the opposite direction and go from low to high. And this is active because it requires energy to put it in the reverse direction. Another way that cells use energy in active transport to control what goes in and out of the cell is a process like this, where say we have a cell and a large molecule that it wants to take inside of it. It can kind of shape around that large molecule like this, because that membrane is very fluid. We described it last lesson with the fluid mosaic model. It's able to bend around that and kind of engulf it until it actually wraps it around completely until it's inside the cell. When we're going in this direction and taking something in, it's called endocytosis. You can think of endo kind of like into. It's taking something into the cell. And it can do the exact opposite. If say this blue material was something you didn't want, then you can go right back in the other direction, start with something inside, and then go from here to here where it's starting to expel it, and here where it's outside. That's when it gets rid of something or something exits the cell. So this is called exocytosis. So that's a look at facilitated diffusion and active transport. Lastly, just to finish this lesson, we're going to look at some applications, and this is going to be looking at kidney dialysis, where let's say the patient has some problems with their kidney, which is basically what filters out the bad stuff in your blood, but unfortunately it's not working for this person, so we're going to use an artificial filter to filter it out. So this isn't going to be like the most medical diagram or explanation of it, but it's going to give an understanding for kind of the basic processes and how we can use passive transport to filter blood. So this is an IV here, and it's going to be taking the person's blood out of their body, and I'm going to draw it in here as having these red circles. Now these aren't representing red blood cells. They're supposed to be representing some toxins, some bad stuff that's in their blood. All right, and this happens to everyone. We just normally are able to filter it out and excrete it, but this person's having a hard time with that. But there's also some good things in their blood. So I'm going to put in green that they have some good chemicals in their blood as well. All right, and then it's coming over to this machine over here, which is our filter, which we're going to use for dialysis. All right, that's kind of like the filtering of the blood here. This is the blood that flows into this side, and what we're going to have over here is called the dialysate, which is what we can control. We're going to put a solution in there, and then we have a semi-permeable membrane here. So I'm just going to draw what we have so far. We have some green molecules, so I'll put 1, 2, 3, 4 there, and we have some red molecules, 1, 2, 3, 4 there. So what we want to do is we want to keep the green molecules in, but we want to get rid of the red molecules. Well, it's because so that the green molecules are not going to want to flow over to here, like we don't want that for the green molecules, we need there to be green molecules on that side so they don't flow from high to low concentration. So let's put some green molecules over there. That's going to prevent us from filtering out the good stuff. Now, do we want to put any red in there? We don't, because we want the red molecules to flow from where there's lots of red ones to where there is less, so to flow over here. And I'm just going to draw a dotted line here to show that once that is kind of filtered out, we're still going to have the green molecules, the good stuff, because they didn't get filtered out, and we're going to have less red. Now, it's important to know we're not going to have none, because this only flows until it's equal. There's 2 on that side, and there's going to be 2 on this side. So it's not gotten rid of all of it, but it's gotten rid of half by just passive transport, no energy. It's just kind of filtering out, and we still have all the green ones that we started with here. So now this part is going to show the blood that's going back into the person, and there is a couple red still. There's about half, but they still have all their green, which is good. That's going back into the person. Now, it hasn't got rid of that much, only about 50%, but we're able to keep this cycle going. We're able to keep filtering the blood through here and here again, because after we have this used dialysate, we're able to just dump it into, like, you can imagine a beaker and get rid of that mixture of greens and reds, and we can have this hooked up to a fresh solution right here of just greens and keep adding fresh dialysate. So we can keep cycling this until we get chopping that bad stuff in half. We lose half of it by diffusion, then half of whatever remaining, and half of whatever remaining, and just repeat this over and over again until the person has very clean blood and they're much healthier for that, and then they don't have to come in for a little while till they get it filtered again. So again, that's a bit of a simplification of the process, but it kind of shows how we can apply passive transport and diffusion to some actual real-life applications. So hopefully that made some sense. Make sure you give the practice questions a try, and I will see you in the next lesson.